We'll kick off our final session. And again, I'd like to welcome Yolanda to talk about the gut microbiome project.
And before Yolanda speaks, we have just prepared 3 slides on the UFI communications on gut microbiome, just to link it to that topic.
Great.
So UFIC has produced a number of materials on the topic of gut microbiome. We have one infographic from 2014, and we have 2 Food Today articles, the latest on a related topic of fermented foods. And as you know, UFIC is leading the dissemination activities of the MinoGut project, a 5-year project. So we produced a number of materials. At the beginning of the project, we introduced what MinoGut's all about in leaflets, UFIC's Food Today article press release. UFIC also contributed to a special issue in the Journal of Trends in Food Science and Technology, which was a special issue dedicated to MinoGut from different partners. We've produced 2 podcasts, one with Yolanda, introducing MinoGut, and we're developing our social media interactions. We're really proud that we've reached over 4,000 followers on Twitter, which is pretty impressive for an EU-funded project. And so this year marks the end of the project, and we'll be communicating, sharing the final outcomes of the research. We'll do this through a press release, leaflet, and UFIC's Food Today article, and we're organising a final conference to take place 18th of October in Brussels. So if anyone here wants to attend, please let us know, and we'll send you an invitation. The conference will be recorded as a webinar with videos of speakers with their slides and made available freely on the website if you can't attend.
One question or a remark. The microbiome is a pretty complicated thing. Is it already up to speak to consumers too?
Well, I think we can discuss about that after the talk of Yolanda.
For UFIC, I mean, communicating to consumers or to scientists. I mean, I still think it's a matter for scientists and not so much for consumers. So,
it's a very good question.
Okay.
But I think let's take it up later.
Okay.
Is the microphone?
Yeah, it's working.
Okay.
Over to you.
Okay. Can I go there?
Sure.
You want to move the microphone?
Yeah, maybe.
Just move the microphone. Should be long enough. It doesn't stay. No, it doesn't stay. You can just let it like that.
Well, good morning, everybody, and thanks so much, Laura, for the kind invitation to participate in the UFIC meeting, and particularly to talk about my favorite topic of research. Well, as you know, the human microbiome has become a hot topic in research due to the fact that a lot of associations have been established in the last decades between alterations in the composition and function of the microbes that inhabit different parts of our body and different disorders. And most of our knowledge are coming from the investigations done initially in the gastrointestinal tract, that is in fact the location of the body where the density of microbes is the highest. And initially the studies were focused on disorders that were located in the gastrointestinal tract, such as inflammatory bowel diseases, and later on gastrointestinal cancer and colon cancer. But later on we have started learning that these alterations in the gut microbiota also influence and connect with other systems and organs, and in fact associations have been established also between what we call intestinal dysbiosis and diseases that have a very different origin, for example type 2 diabetes or type 1 diabetes. In fact, nowadays we talk about the existence of a gut-brain axis whereby the microbiota of the gastrointestinal tract communicate with the brain and can affect behavior and mood, for example. And we think that also can play a role in the development of psychiatric disorders and neurodegenerative disorders. We also talk about existence of a gut-liver axis. This is a bit more obvious because there is a direct connection between the gut and the liver via the portal vein, through which metabolites from the gut microbiota impact on the brain directly. And we have started talking about a connection between the gut and the lung, probably because there is a connection between the immune system of all of our mucous and the microbes that inhabit our mucous. And well, this in fact has changed completely the way we investigate the origin of disorders, different disorders, and the way we are envisaging the possibilities to prevent and to treat disease. And this affects the nutrition and the health area as well. Now we consider that the microbiome contributes to all the aspects of the diseases, including the inter-individual variability. This means that the microbiome can contribute to our vulnerability to disease, but also to the disease prognosis and the efficacy of the treatments. And on the other hand, well, on the one hand, this adds complexity to the problem because we need to consider a new actor in all this process. But on the other hand, it gives also us the opportunity to interfere with the development of the disease and perhaps prevent the development of disorders using also strategies that targeting the gut microbiome can modulate the function of our microbes and in this way steer in another direction this path from health to disease. And one of the alternatives that have been proven to work are the fecal transplantation trials that work with Clostridium difficile infection. This is only done at experimental level in the cases that cannot be treated in another way. It's not a very refined way of treating this disorder because we don't control all aspects of the safety of these fecal transplantation experiments, but we hope to be able to progress into more refined strategies, selecting bacteria that can contribute to a healthy microbiota in an overall context, and also the use of dietary strategies to modulate the function of the gut microbiota is one of the most straightforward alternatives that we have. And well, in the rest of my talk, I'm going to talk about obesity because, as you know, it's one of the main health issues. But despite the fact all health policy measures that have been put in place, no country has reversed this epidemic yet. And we have been recalled the need of providing new thinking in this area to try to understand better how the environmental factors interact with our biological determinants of the vulnerability to develop obesity, to try to find out better methods to prevent these disorders. And from our point of view, the investigations on the gut microbiota constitute part of this new thinking. Well, in relation to the theories that have been proposed to explain the obesity epidemic, one of the oldest ones is our adaptation to safe energy, an ability that can help us to survive in times of food scarcity, but that can contribute to obesity in times of food abundance. And I wanted to mention this whole theory because it fits very well with the first role that was attributed to the gut microbiota in the context of obesity. Well, initially, probably most of you know this paper that was published in 2014 and impacted all of us a lot because they compare germ-free mice with colonized mice, and they observed that colonized mice has increased adiposity and developed insulin resistance. So from these observations, it was concluded that overall the colonization of our gut with the microbiota contribute to the obesity phenomenon. And this was explained via mechanistic studies done in rodents mainly, where it was demonstrated that the gut microbiota contribute to extract energy from the diet, particularly from complex polysaccharides. This was translated in an increase in energy metabolites in the intestinal lumen, but also in peripheral blood. And this was also translated in changes in the expression of genes that overall contribute to the storage of lipids in the liver mainly, also in the muscle and in the adipose tissue. And well, in fact, one of the properties that have been investigated in more detail is the ability of our gut microbes to utilize complex polysaccharides, fibers, because they are specialized on this activity, because in fact fibers are the main energy source for our microbes. And through these complex metabolic reactions, different short chain fatty acids are generated, mainly butyrate, propionate, and acetate, also gas, whose utilization by methanogenic and apoptogenic bacteria increase the fermentation rate. And it has been estimated that overall this activity of our gut microbes can provide 10% extra energy daily, something that in the long term can contribute to obesity. But well, despite the fact this is from a biochemical point of view plausible, we have to take into account also that the energy that is provided by simple sugars in comparison with the energy provided by fibers. So this theory in principle contradicts to the beneficial effect attributed to fibers in weight management, and according to the calories provided, it's logical to think that it's better to replace simple sugars with fibers. And we have also learned that some of these short chain fatty acids generated by the gut microbes during this fermentation process can play a beneficial role in the context of obesity. One of the metabolites better investigate is butyrate, that we know that feed the colonocytes and regulate the production of incretins, such as glucagon-like peptide 1 that induce satiety, or glucagon-like peptide 2 that strengthen the gut barrier function. It can also contribute to develop an anti-inflammatory role via inhibition of PPAR gamma and the inflammatory cascade mediated by the nuclear factor kappa B, and also via epigenetic regulatory mechanisms. And propionate has also been investigated with similar functions, and in fact recent publications of studies in humans using inulin propionate that release propionate in the gut have been demonstrated that reduce energy intake, and this is associated with an increase in the production of glucagon-like peptide that induce satiety. So in general now we think that this first theory was a bit simplistic about the role that the microbiota could play and the origin of obesity, and I would like to, or I'm more in favor of this second theory, that is the maladaptation to an excess of energy that could have caused alterations, profound alterations in the composition of our gut microbiota, what we call dysbiosis, that in turn aggravate the adverse effects of unhealthy diets on our metabolism. And well, I would like to mention three studies that exemplify how this theory can work. Well, this is a study that was published in 2009, and the authors demonstrate that comparing germ-free mice and colonized mice, that when these mice were fed a Western diet, those that were colonized with microbes show increases in inflammatory factors, but not those that were germ-free. So it was concluded that was the interaction between the microbes and the diet, the one that drives the inflammatory process underlying obesity and causing the chronic disorders such as type 2 diabetes. And well, it was concluded that the Western diets favor the increases in gram-negative bacteria that provide LPS and could signal via toll-like receptors and activate this inflammatory cascade. This is another example of another mechanism by which this could work. In this paper, the authors demonstrated comparing wild-type mice and the same mice fed a high-fat diet that developed obesity, that the high-fat diet reduced the expression of antimicrobial peptides produced by the host, the REC3 gamma peptide. This leads to dysbiosis, and when they transfer the dysbiotic microbiota of these mice into new recipients, this microbiota contribute to increased body weight gain, showing also causality. So the microbiota was at least partially responsible of this increase in body weight.
Yolanda, What is the role of MIT88? What does it do?
This is an adapter protein that is in the signaling pathway of toll-like receptors that are one of the innate immune receptors that recognize bacterial motifs such as the lipopolysaccharide.
And what are the features of the knockouts? How are they different in their metabolism?
It depends. Well, the MID88 protein is an adapter that works, that connects several toll-like receptors. So it's not that this mutant is very specific.
Okay.
Of one or,
so you mean, notice differences only if challenged by different diets. You
yeah,
notice
yeah.
on Adlebiton.
I will tell you that, later on that in the case of the knockout, the toll-like receptor 5 knockout, there is a specific phenotype. The animals develop hyperphagia and obesity and metabolic syndrome spontaneously without any challenge with the diet. But this adapter is really a molecule that is in the middle and it's a bit unspecific. And not all type receptors play the same role in obesity. And well, this is another example of this type of theory. In this case, the authors compare the effects of a diet rich in PUFAs and a diet rich in saturated fat. And in the context of this diet, what happens is that the microbiota chain, there was a bloom of delta proteobacteria, particularly a bacteria that is called Bilobia wasworthia, that is able to survive in the presence of bile acids conjugated with taurine. And these bacteria produce hydrogen sulfate that is toxic for coronocytes and cause inflammation. And well, I must say that this was initially tested in animal models, but later on it was also demonstrated in humans that were switched from a normal diet to a high fat diet, that they also have increases in Bilobia wasworthia and alterations in bile acid metabolites. But well, one of our major challenges, even if we have started understanding how the microbes can contribute to obesity through experiments done in animal models, is to understand which bacteria or consortium of bacteria play a role in human obesity and which ones can help us to combat obesity. And well, to address this question, many observational studies have been done comparing the microbiota of lean or obese subjects or following up the microbiota of obese subjects that were under dietary or lifestyle intervention to reduce body weight. And in many of the studies, what was shown is that the increases in firmicutes, or subgroups of firmicutes, were related to an obese phenotype, and increases in bacteroidetes or bacteroidetes were related to a lean phenotype, and also the reduction of body weight was related to increases in bacteroidetes. However, we should admit that these conclusions were also opposite to publications that were published in the last years. And for example, this is a publication of the MetaHit group that include a relatively large number of subjects, more than 100, and they subdivide the subjects in two groups, those that have low bacterial gene counts and those that have high bacterial gene counts. And those that have high bacterial gene counts were those that more often have metabolic dysfunction, and in those subjects, the abundance of bacteroides and some other bacteria was increased. So the associations were opposite to the ones I showed you before. Well, when we started the MyNuGat project, we were more or less at this point. And the fact is that even if these studies have been published in very high-grade journals such as Nature or Science, the situation is that in most of the cases they were not controlling for confounders such as the diet or physical activity or medication. And now we know that many of these associations are completely biased by the confounders. So in order to shed some light into this area, we initiated a European project 4 years ago. We are in the 5th year of the project to try to understand better which could be the role of the microbiome in energy balance. And we have also worked on the role of the microbiome in brain development and function to try to use this information to tackle diet-related diseases and behavioral disorders. Well, these are the main 5 objectives of our project. First of all, we wanted to understand better which microbes contribute to nutrient metabolism and which are the effects on human metabolism. We wanted also to identify possible microbiome-related futures that could predict obesity. Also identify the role of the gut microbiota and metabolites in eating behavior and emotional behavior. Also, we have worked with cohorts that were followed up at very early stages in life to understand better how environmental factors and lifestyle habits could impact on the microbes and could also impact on the brain, immune, and metabolic health programming. And the last objective was to develop new foods and ingredients, including probiotics that could reduce the risk of metabolic disorders. Well, I will tell you only about a few aspects related to these objectives because we don't have the whole morning for that. But I would like to tell you about the results we have got in one of the studies where we have investigated the role of the microbiota in nutrient metabolism and energy balance. And well, we did a study to evaluate the effect of dietary protein, first because the intake of protein in Western countries is higher than the recommended amount on average. Also because high protein diets are usually used to reduce and to control body weight, and these diets consist of from 2 to 3 times the recommended daily intakes. We know that high protein diets have beneficial effects in terms of satiety, also modify beneficially the lipid profile, and contribute to weight reduction, but they're also associated with negative effects on kidney function and also are related to intestinal inflammation and cancer. However, despite the fact these diets are used very often, we don't have a maximum non-deleterious intake level for proteins yet. And also, we have little information about the role of the microbiota in all these transformations of the part of protein that reaches the large intestine and which is the contribution of gut microbes to beneficial or detrimental effects. So as you know, proteins are digested in the upper part of the gastrointestinal tract. In general, high protein diets have beneficial effects in terms of metabolism, but a large proportion of the proteins also reach the large intestine. And we know here that there are a lot of metabolic reactions, mainly involving the fermentation of free amino acids. And we wanted first to establish which could be the benefits and risks of these diets. And another question we wanted to address is whether the source of protein also matters, not only the quantity. And we choose casein and soy because, well, these represent 2 types of protein, one from animal origin and the other one from plant origin. We know that there are differences in amino acid composition. For example, casein proteins have increased proportions of cysteine, lysine, and leucine, or soy protein have increased proportions of arginine, phenylalanine, and glutamic acid. And we also know that these proteins show different digestibility. The digestibility of soy proteins is lower than the digestibility of casein proteins. So we were wondering whether this difference will also make a difference in the activities that the microbiome could develop. Well, to test this hypothesis, we ran a double-blind randomized controlled trial with 3 arms. One received a placebo that was maltodextrin, the other one casein, that represent 15% of the proportion of protein, that in total was 30%, and the other arm consumed soy as main source of protein. Can you please repeat what the placebo group received? Maltodextrin. Maltodextrin. Thank you. When the trial lasts for 3 weeks, and we took different samples at 4 time points, stool, urine, and blood, and we also took rectal biopsies at the beginning and at the end of the trial. We checked compliance by measuring blood urea, and compliance was very good. We also check the dietary records, the food intake, particularly not only to check compliance, but also to see if the dietary patterns were changing, and this could be a confounder. Fortunately, there were no difference in fiber, so that is the nutrient that could affect more changes in microbes. And well, in relation to parameters that are associated with metabolic health, we didn't observe a significant difference, mainly due to the fact that the diets were isocaloric. We did it like that because otherwise we would have been investigating the effect of the energy content of the diet, not the effect of the source of protein. That was our objective. And the only difference we found was a reduction in systolic blood pressure in those subjects that consumed the soy protein, but perhaps this could be due to the fact that isoflavones were still linked to the isolate protein. Well, what was more interesting to us is that the fecal metabolome was changing. Was changing as a consequence of the amount of protein, but also as a consequence of the source of protein. And well, in those cases, when we increased the proportion of protein, butyrate was reduced. That is mainly coming from carbohydrates, but other metabolites were increased, such as branched-chain amino acids, leucine, valine, isoleucine, that has been related to the development of insulin resistance, for example. Tyrosine was also increased, and also some other branched-chain compounds. Well, in the case of the soy protein, we found decreases in butyrate and increases in tyrosine too, but also other changes that were not common to the changes observed with casein, such as increases in phenylalanine, valerate of tyramine. And we also found few changes comparing the two high protein diets. Well, another thing that was interesting was also to look into the urinary metabolome, because we also found difference as a consequence of the amount of protein. We observe increases in isobutyrate, phenylacetylglutamine, that are not very nice compounds for our body, and indoxyl sulfate, that is a kidney toxin. And but well, we also observe difference depending on the type of protein, because paracresyl sulfate that comes from paracresol that is generated by the microbes was increased in the subjects that have the high casein diet, and hydroxyphenylpropionate was increased in those that have the high soy protein diet. So we conclude that not only the quantity of protein, but also the quality of protein matters. In terms of changes in the microbiota, there were not too many changes. I want to highlight the increases in roseburia in those subjects that consume the high soy protein diet, because roseburia is a butyrate producer, and reductions in the alister in those that consume the soy diet in relation to those that consume the placebo. But well, the most relevant changes were in metabolites that were partially produced by the microbes. And well, what we found interesting was also to identify correlations between specific metabolites and specific bacterial taxa that were quite consistent with the literature. For example, butyrate was correlated with bacteria that are known that are butyrate producers, and some other potential toxic compounds, such as paracresol, was correlated with oscillospirine. And well, these type of correlations could be tracked later on in functional studies to corroborate the biological value of such statistical findings.
Can you go back to the slide before?
Yes.
You have a lot of variability.
Yeah. The main source of variability, the main difference are between the individuals.
Yes. So I was wondering, okay, we'll ask question again. Okay, I just wanted to make sure
No, you can do it. No.
that I saw the variability.
No, the variability is amazing, and this study was a relatively small scale, because we have something less than 50 subjects per group. but it's also very challenging to have volunteers to do this type of trials. And this is always the problem. Also was short term, because in 3 weeks you cannot see too much.
What was the age range of the people?
Were adults.
From
I don't
19.
remember adults. Were overweight.
So the diabetes is not unusual, but I was wondering what would be your opinion if this protocol was not done on parallel, but was a crossover with a proper washout period?
Well, this is another alternative to reduce variability, but the problem you usually have there, you need to engage the subjects for longer time, something that is not easy either. And another point is that you have to be completely sure that the washout is enough to continue the trial and you don't have carryover effects. So there are pros and cons in the two types of designs.
And what would be the washout time?
Well, we did an intervention with PUFAS and fiber, and we have a washout period of 1 month, and we are not completely convinced that was enough in this other intervention that we did with crossover design.
Yeah, in some publications I read they would let people wait for 3 months to ensure the full recovery of the gut flora. Yeah. That would be,
It's a bit complex
yeah.
practice.
Losing half a year study.
Well, as we identified some toxic compounds in the metabolomic studies, we wanted to see whether these fecal extracts could exert any cytotoxic effect, and we did it in cell cultures, exposing the cell cultures to these fecal extracts, and we could not observe any cytotoxic effect. Again, the main factor of variability was the individual. We could classify, in fact, the samples into those that have low cytotoxicity potential and those that have high cytotoxicity potential, but we could not assign the samples to the dietary pattern. But what we observed were difference in the transcriptome, in the colonic biopsies, and we observed that those samples, the differential gene expression, when comparing samples before and after the diets, was significantly modified in those subjects that have these high protein diets. And also, when we look at the functional pathways these genes were belonging to, we observe difference between the two types of proteins, between those that ingest soy protein and those that ingest casein. And these differences were affecting pathways related to cell death and survival, cell signaling, or the cell cycle. Well, another objective we have had in the project was to try to identify microbiome-related features that could help us to predict the development of obesity. And well, this was quite challenging because to do so, we need to do long-term longitudinal studies that in the time frame of a project is quite complicated. And while we took advantage of the IDEFIX type family cohort that is led by Wolfgang Harens, well, these are 2 European projects, and we took advantage of the fact that they collect some samples in 2009 and 2010, and during the MinuGut project we collect another pool of samples in 2013 and 14, and from these samples and subjects we were able to identify some subjects that remain normal weight and some infants, some children that develop overweight during the time course. Well, the most interesting findings are the following ones. Doing clustering analysis of the subjects according to their microbiota, we could identify 4 different clusters. C1, C2 that correspond to the children that were normal weight, and C3 and C4 that correspond to the children that were obese, overweight. And well,
Sorry, Yolanda, so you got the clusters So you got the clusters based on the gene expression analysis? Or
No, we made the clusters on the microbiome of the
oh, the
sapiens.
microbiome, and they clustered according to the weights that
Yes.
you kind of analyzed. So you got the clusters fitting into those.
Exactly.
Oh, wonderful.
Yes, wonderful. It's a concept similar to the enterotypes concept. I suppose you have heard about it. Well, here what we look also is into the possible relationship between these clusters related to the microbiota and the obese or lean phenotype and the inflammatory markers. And we observe also that cluster 3 and 4, particularly cluster 4, was associated with inflammatory markers in these children that were also obese, and also with some metabolic markers such as increases in triglycerides or diastolic blood pressure. And we also follow the dietary patterns of these children, and what we found also is an association between the clusters C3, C4 related to the microbiota and the dietary patterns D2 and D5 that are characterized by increases in intake of carbohydrates and fat and increases in the intake of protein and high fat. And well, this is associated with the obese phenotype, and what's interesting to us is that this combination between the microbiome clusters and dietary patterns was also present in those children that were normal weight initially but developed overweight in the time course. So we conclude that perhaps this microbiome configuration can be one of the predictors of the development of obesity, of course, together with some other factors such as genetics, physical activity, and many other variables that we know influence the development of obesity. But this is our theory. And well, finally, as I told you, we are also working on the development of foods and food ingredients that can help us to attenuate and reduce the risk of developing metabolic disorders. And in particular, we are working on the development of a next generation of probiotics that perhaps can be more efficient than the classical probiotic ones. And when we start working with bacteroides, due to the fact that in the literature initially bacteroides were associated with a lean phenotype, although, as I told you, there are controversies. And what we did was to screen bacteroides that we isolate in our laboratory in cell cultures to evaluate the pro or anti-inflammatory properties in macrophages. We select the Bacteroides uniformis strain because this was the one that induced the most promising anti-inflammatory profile in vitro cultures. And we also, looking at our own studies, epidemiological ones, we observed that the Bacteroides uniformis was promoted, was associated with breastfeeding in a study that we did in infants. And as you know, breastfeeding protects against the development of obesity and type 2 diabetes. So in order to understand whether these bacteria could help preventing or treating obesity, we did just a study in mice. Mice fed a normal diet. A group was fed with placebo, another group Bacteroides uniformis, and mice fed a high-fat diet to induce obesity, one fed placebo and one Bacteroides uniformis, and we analyzed metabolic parameters and immune parameters. Well, as was expected, the mice that consumed the high-fat diet show an increased body weight gain in comparison with those that received the normal diet. Also, an increase in the adipose tissue, also show an increase in the area under the curve of glucose after an oral challenge of glucose, and also develop hyperleptinemia, hyperglycemia, hypercholesterolemia, and hyperglyceridemia. But when we include the bacteroides in the diet together with the high-fat diet, we could reverse all these metabolic alterations and also observe reductions in body weight and in fat mass. And well, we wanted to understand which could be the possible mechanisms of action, focusing on anti-inflammatory mechanisms. And well, what we did is to analyze different cellular populations in different compartments, in blood, in player patches, and adipose tissue. And for example, we observed changes in cytotoxic cells. In the 3 compartments, increases in B cells. Also, in the adipose tissue and in the player patches, we observed increases in pro-inflammatory macrophages. And also in the 3 compartments, we observed reductions in T regulatory cells, and these changes were reversed by the bacteroides. We also look into the effects on inflammatory cytokines and anti-inflammatory cytokines, and well, we observed that the high-fat diet increased the production of interferon gamma in the 3 compartments, also other inflammatory markers in peripheral blood, while reduced the production of anti-inflammatory cytokines such as IL-10 and IL-33, while the bacteroides reverse most of these adverse changes. And well, we wanted to understand possible molecules involved in this process, and we analyzed also the expression of toll-like receptors. And what we observe is the high-fat diet reduced the expression of these 3 toll-like receptors, but particularly TLR5 in payer patches, but also in the adipose tissue. While in the presence of the bacteroides, the expression of toll-like receptor 5 was increased. And well, as bacteroides is not a producer of flagellin, that is the bacterial component that activates toll-like receptor 5, we did some experiments in cell cultures to understand if this was a direct effect of bacteroides or was an indirect effect because bacteroides could modify the microbiota and then these changes could activate the expression of toll-like receptor 5. So we did an experiment in cultures exposing the cell cultures to the fecal samples that represent the overall microbiota composition, and the fecal samples of the obese mice reduced the expression of toll-like receptor 5, while the microbiota of the mice that received the bacteroid increased expression. And we wanted to go beyond that and understand whether the bacteria itself could activate toll-like receptor 5, and to do so we did experiments in cell cultures, but using pure cultures of the bacteria. So in these experiments, we demonstrate that the bacteria, that bacteroides as such, can activate toll-like receptor 5, and this is a positive control that is flagellin.
And that would be human gut cells, like or
Sorry?
So the cell culture, which cells did you use?
Well, these are kidney cells that overexpress the toll-like receptor 5.
Okay.
It's a model system,
Okay.
artificial one.
Okay.
It has a reporter and you can measure the activation of toll-like receptor 5 in a very easy way. I think that the measure is colorimetric.
But they are of human origin, the kidney cells, not of mouse origin.
I don't know exactly, probably. But well, I think that the origin of the cells is not so important because you have an overexpressed gene that has a reporter. So it's an artificial system in what I mean.
Excuse me, how long does it take for you to reverse the obese phenotype into a lean one?
Well, we run the experiment for 40 weeks, but you can have an obese phenotype in 7 weeks.
Mm-hmm.
The point is that if you want to investigate the inflammatory process in peripheral tissues, you need longer time.
Mm-hmm. And from the obese to the lean?
We were doing it in parallel, so
mm-hmm.
we were feeding the high-fat diet together with the bacteria.
Mm-hmm. And how long did it take?
To see changes in reductions in body weight after the 7 week for sure.
I see. Mm-hmm.
But it depends a bit on the batch of animals because I mean the evolution of body weight is not very homogeneous.
Yeah, you have lots of variability between individuals. Yes. Okay.
That's why the ideal situation is to test all that you have to test in the same batch of animals and at the same time. Well, we look at the composition of the gut microbiota, as a consequence of the administration of the high-fat diet and the Bacteroides uniformis, and we observed changes in beta diversity, mainly due to the diet, was the main factor contributing to these changes, but secondly also due to the administration of the Bacteroides, and from the point of view of difference in taxonomic groups, what we observed were increases in Helicobacter due to the high-fat diet and reductions due to the administration of the Bacteroides. So in summary, I'm going to finish, don't worry. The high-fat diet contributes to intestinal dysbiosis, reducing the presence of Bacteroides and increasing the presence of Helicobacter. This changed the expression of TLR5 mainly, and this causes inflammation in peripheral tissues and systemically. And this leads to alterations in the metabolic phenotype, for example, intolerance to glucose, and also development of obesity. And well, when we administer the Bacteroides, there is a restoration, partial restoration of the gastrointestinal ecosystem, increases in the expression of toll-like receptor 5, and this is translated into a reduction of the pro-inflammatory phenotype and a restoration of glucose tolerance. And well, we speculate that this is happening through toll-like receptor 5 signaling, because as I told you, the knockout in toll-like receptor 5 develop hyperphagia and metabolic syndrome in a spontaneous manner. So in summary, we can say that diet is a major driver of gut microbiota composition, and therefore is instrumental to modify our health. In our case, what we have proven is that it can impact metabolic health and its relation to obesity. We can also conclude that not only the amount of protein of the diet is important, but also the quality of the protein, and that depending on that, we can modify effects in the mucosa gene expression that can impact in the long term on our gut health, and also microbial related metabolites that can be involved in these effects, and this should be considered when evaluating the long-term effects of dietary proteins, and particularly the high-protein diets. We also consider that the diet-driven microbiome configuration may help to predict obesity. And while we are on the way to prove that some indigenous intestinal bacteria, others like classical probiotics, can constitute the next generation of probiotics with enhanced efficacy against metabolic and immune alterations related to healthy diets, and that at least the bacteria we have evaluated could operate and act via anti-inflammatory mechanisms that may be signaled via the toll-like receptor 5. And that's it. Thank you very much for your attention.
These are
all the collaborators that have participated in the results I presented.
Thanks very much for that really fascinating presentation.
Yes.
And lots of new information there that I certainly haven't come across before. Just a very quick question, and then we'll open the floor. If we were to deliver these bacteria, the bacteriologies, let's say in the diet, how could you protect them so they could get down into the gut and have the beneficial action?
Well, that's a good question, because these bacteria are anaerobic, and the production at industrial scale is quite challenging. And I can imagine that the passage through the gastrointestinal tract is also another hurdle that they have to survive. There are encapsulation technologies that are being developed very fast, but we have not yet tested with this bacteria what we can do.
So we've done a few studies on animals, probiotics in animals, on weight management and inflammation, and unfortunately, when we with beautiful results, with absolutely amazing results, and then when we went to the human clinical trials, it really we did not see at all the same results. So my question to you is, what do you think are the criteria that we should take into account when when we move from that phase of animal to human when it comes to the effect of probiotics um, in health, especially in the metabolic um, area?
Well, it's difficult to talk about unique criteria that you have to follow in order to succeed. But it's true that it's difficult to think that with only one bacteria at the doses that the bacteria administer are going to have an important effect in relatively short time, because the studies are not longer than 3 months.
We did 6 months.
6 months, that's a lot. But well, in general, I think that the doses that are being used are probably not enough to be effective. And on the other hand, perhaps we need to integrate other components. I mean, perhaps unibacteria is not going to do all what we need.
Yeah, we also did. Okay, it's I understand it's a complex, but we also did the combination with calorie restriction, and I'm just wondering how much correlation there is between animal studies and human studies in this area, and I don't see much in the literature, so maybe there's something that we're missing and we're not seeing that I thought you might have insight.
Well, there are some publications, I think that at least 3 of, I think that Lactobacillus reuteri, that was tested in animal models, was working, and then they did the trials in humans, and it works. I don't remember exactly the quality of the studies.
At least this is and one in Canada.
At least this is an example that has worked. But yeah, I agree that there are many others that have not worked or not as well as expected. But I think that the strategy should be an integrated strategy. I think that if we think of how to tackle obesity, we cannot focus on a bacteria and a probiotic, or, I mean, this is not realistic. Well, for example, with our bacteria, what we are doing is we have tested different fibers to see, because this is a bacteria that is in most of our intestines, and it's specialized in the utilization of complex polysaccharides. What we have done is to test in vitro which polysaccharides the bacteria can use to promote its growth also, and what we are doing is trials with both the bacteria and the fibers that work better, and the idea is to have such kind of approach that I think will give us more opportunities of succeeding.
I do have a technical question, and this goes back to my own involvement in gut flora studies in infants, and that's how it was called 30 years ago, gut flora and not microbiome. So, and I remember the difficulties in feces sampling, in differentiation between anaerobes and aerobes in the feces, and how to store, and how is it done today to differentiate the gut flora or the microbiome, to differentiate between the individual bugs coming up, like the bacilli, bacteroides, steroides, or whatever. How is it done today?
Well, now we use DNA sequencing, so there are no such huge problems as at the beginning to characterize a microbiota. It's relatively straightforward. Anyway, we still have a lot of challenge because the methodology is not standardized. Everybody does what is working in his own lab, and this is a problem. And also it's true that, I mean, we don't have too many troubles in the sense that we can really cover a wide range of taxonomic groups, and we can analyze all in one run. The point is that the taxonomic resolution, for example, is still very poor to give a meaning to our results.
If you're using whole genome sequencing, you can fish the DNA out of the gut, and you can classify the bacteria accordingly.
So you don't have to grow them anymore. You don't grow them.
I understand this, but I remember probably with feces sampling, and then we had it, if you analyzed it directly, you had a different composition than 8 weeks later, and so...
No, that's still the case. I mean, the way you collect your samples, the way you store the samples, the time that has passed between the moment you store the samples and when you analyze them has an impact. And that's why we are trying to establish some standards, at least minimal standards, that all of us can follow to be able to do cross comparison, something that now is really almost impossible. Well, we do it, but all these comparisons are biased.
We were actually trying 30 years ago the influence of lactoferrin on the gut flora of babies, and didn't really see the results we wanted to see as a baby food gut.
But I wonder, Yolanda, if you freeze them in liquid nitrogen and do the sequencing afterwards, the DNA stays in there, right?
Yeah.
And stays intact, and you should be able to differentiate between the taxonomic groups regardless. I mean, you should be able to identify the species in there.
Yeah.
We can, sure.
Yeah.
There's some colleagues in Denmark, and they're taking feces from all over the world, and they're taking samples and they're analysing for different pathogenic groups and looking which pathogens are established in different areas of the world.
That's a whole project.
And one or two countries actually refused to send them samples. So what they did was they sampled the contents of the airplane when it landed. So science is, you have to expect that person. No, that is fascinating. And you're using, you're actually fishing out the DNA and you're analyzing that way rather than fishing out and so on.
Yeah, but the DNA in terms of determining the species, that's fine. But then indeed the metabolome may differ, right? Depending on the storing sample, on the storing of the sample.
Well, the metagenome can also differ depending on the way you store your samples. It's not that everybody is doing it in the same way, and it's not always possible to do it in the same way. So.
Can you give us a little insight into into behavior and the effect of gut microbiota on the behavior?
Well, the experiments we have done have been mainly on animal models. I don't know where Christine, I think that was in our last meeting, I know about it. What has been done so far by a group that is in Austria, Peter Holzer, is reproduce a phenotype that is a depressive-like phenotype in this obese model. And they have also analyzed metabolites, but I don't remember exactly if they have finished. Well, they established some connections between alterations in the gut microbiota and this depressive-like phenotype, and they have also done metabolomic analysis in the brain and in different tissues. And well, we have done also some trials, and we relate this depressive-like phenotype also with toll-like receptor 2 signaling in the gut and in the hippocampus. But this experiment was done with a bifidobacteria, and I then wanted to put it here because it was going to disturb you a bit if I start talking about bacteroides and bifidobacteria in the middle.
I mean, in terms of the work of UFIC and for dissemination of information, or what could you say you have at the present moment that would be useful for UFIC to disseminate? What do you have? What results would you have at the present moment that would be useful for UFIC in terms of dissemination?
Well, I think we have already disseminated part of the results that we have now. I don't know if you want to elaborate a bit more, we could do it.
I would say, I mean, in terms of, if you look at the consumers, I mean, what would be the advice or the status of science that we can transfer in terms of practical advice on things that people can do? And I guess that is still sticking to a healthy, balanced diet, probably.
Yeah.
For the time being.
For the time being. I think that the message about the effect of proteins can be communicated to consumers easily. I think this is a very important message. And also the results that are coming from the fiber interventions can be communicated to consumers too. I have not presented the results here because we are still integrating the metabolomic data also. And the main intervention, that is the longer one, and we will have the strongest results, is now finalizing, so we will have the results at the end. But I think that the relation between the diet, the microbes, this is really very easy to understand, and I think can be useful for the consumer too.
Are there studies done on, I mean, you mentioned the high protein diets, but the carbohydrates were still quite high. I mean, have you or are you aware of studies that look at high protein, high carb diets and very low carbohydrates, so the ketogenic, for example, diets?
Well, this is in literature, there are studies with this type of design.
Yolanda, you mentioned it's not only the quantity, it's also the quality of protein. But then what would be, so if you compare casein versus soy protein, what would be your conclusion? What would be the beneficial source of protein?
Well, we cannot say that there is a winner and there is a loser. So that's the truth. But well, we have the impression that in the context of the casein diet, for example, you have this paracrysyl sulfate that you don't detect with the soy protein diet. And with the soy protein diet, you have increases in roseburia that produce butyrate that perhaps attenuate the adverse effects of high protein diets on the production of these beneficial metabolites. So I tend to think that the soy protein perhaps exert less adverse effects.
But I don't think that's at the level that can be.
A new issue for Susan coming up already, you publish something and then all of a sudden the soy eaters are getting 20 years older or whatever.
And regarding aromatic amino acids and the release in the gut, are there any studies running regarding gut-brain gases and neurotransmitters?
Well, there is in the animal studies we are not working exactly on that because was Peter focusing on this obesity model. In the relation to the gut-brain axis, what has been done is a study in humans. Well, there are a couple of studies, one is still running. The one that has finished, they have evaluated the possible role of the type of birth that in theory modifies the gut microbiota in the response to stress. And what they observe is that depending on the type of birth, the response to stress changes and the inflammatory markers changes in adulthood.
Thank you very much. We should go a line on the discussion then unless there's any really pressing question. No, thanks very much for that. It's been fascinating.